The kidneys play a very vital role. They filter blood by removing nitrogenous wastes, regulate blood pressure, and assist in the production of red blood cells. The kidneys are compact, bean-shaped organs on either side of the spinal cord in the lower back. More precisely, they are situated between the last thoracic and third lumbar vertebrae in close proximity to the dorsal inner wall of the abdominal cavity. The 11th and 12th ribs partially protect the upper part of the kidneys. The right kidney, which is near the liver, is slightly lower than the left one, which is adjacent to the spleen. The kidneys are reddish-brown in color. In an adult, each kidney measures about 10 to 12 centimeters in length, 5 to 7 centimeters in breadth, and 2 to 3 centimeters in thickness. Each kidney weighs about 120 to 170 grams. The kidneys have an outer convex surface and an indentation or a notch on the inner concave surface called the hilum, through which blood vessels, nerves, and the ureters pass. The kidney has a tough, fibrous outer layer called a capsule, and its longitudinal section shows that its inside is differentiated into two main regions, an outer cortex and an inner medulla. There are about 8 to 18 cone-shaped structures in the medulla called medullary pyramids, which project into the calyces. The tip of each medullary pyramid is called a papilla. The cortex protrudes or extends between the pyramids as renal columns called the columns of Bertini. The medullary pyramids project into the calyces, which lead to a wide funnel-shaped space called the renal pelvis. Each kidney contains about 1 million nephrons in the cortical region, which are the basic functional units of the kidney. The nephrons filter nitrogenous wastes and excess salts from the blood and help in body fluid regulation and urine formation. Each nephron has two parts, the glomerulus and the renal tubule. The glomerulus is a ball-shaped network of capillaries formed by the afferent arteriole, which is a fine branch of the renal artery. It performs the first step in filtering blood. The filtered blood from the glomerulus exits through the efferent arteriole. The other part of the nephron, the renal tubule, helps in the reabsorption of salts and water as the urine passes through it, thereby concentrating the urine. The renal tubule begins with the Bowman's capsule and consists of proximal convoluted tubule PCT, Henle's loop, and distal convoluted tubule or DCT. The Bowman's capsule is a double-walled funnel-shaped structure that encloses the glomerulus. It collects the glomerular filtrate. The Bowman's capsule, together with the glomerulus, is called the Malpighian body or renal corpuscle. The Bowman's capsule opens into the proximal convoluted tubule, a highly coiled structure, and is followed by a hairpin-shaped loop called Henle's loop. Henle's loop has a descending limb and an ascending limb. The ascending limb opens into the distal convoluted tubule, which is also a highly coiled tubular structure. The distal convoluted tubules of several nephrons open into a collecting duct. Several collecting ducts converge and pass through the medulla into the renal pelvis, which leads to the ureter. The collecting ducts, along with Henle's loop, lie in the medulla, while the Malpighian body proximal convoluted tubule, and distal convoluted tubule of a nephron lie in the cortex of the kidney. The nephrons in the kidney are also distinguished as cortical nephrons and juxtamedullary nephrons based on the length of Henle's loop. Cortical nephrons constitute the majority of nephrons and have a very short Henle's loop that just about extends into the medulla, while the juxtamedullary nephrons have a very long Henle's loop that runs deep into the medulla. Each nephron also has a fine network of capillaries called Peritubular Capillaries, which is formed from the efferent arteriole leaving the glomerulus. A minute vessel from this network of capillaries forms a U-shaped structure called the vasa recta, which lies parallel to the loop of Henle. They are greatly reduced or absent in cortical nephrons. The capillaries of many such efferent arterioles unite to form the renal vein, which joins the inferior vena cava. Let's now study the other parts of the urinary system. The kidney opens into the ureters through the renal pelvis. Each ureter is about 25 centimeters long and carries urine from the pelvis of the kidneys to the urinary bladder. The urinary bladder is a hollow, muscular and elastic organ that temporarily stores urine till it is released from the body. Urine leaves the urinary bladder via the urethra, a tube through which urine is discharged. The urethra is enveloped by the urethral sphincter, which controls the flow of urine from the bladder to the outside. Hence, the urethra, along with the urinary bladder, ureters and kidneys, constitute the human excretory system. The kidney is the major excretory organ in humans, and one of its primary functions is urine formation. Each kidney contains about 1 million nephrons, which are the basic functional units of the kidney, and help in urine formation. Urine formation involves 3 major processes, namely glomerular filtration or ultrafiltration, reabsorption, and secretion. The first step in urine formation is the filtration of blood, which takes place in the glomerulus, and hence the name glomerular filtration. The pressure of blood in the glomerular capillaries forces the blood to pass through 3 layers, namely the endothelium of the blood vessel or the capillary, the basement membrane of the glomerulus, and the epithelium of Bowman's capsule. The epithelial cells in Bowman's capsule, also known as podocytes, are intricately arranged, leaving a few minute openings called slit pores or filtration slits. These slits or membranes help in the filtration of blood, and almost all the constituents of the plasma except the proteins pass into the lumen of Bowman's capsule, a reason why glomerular filtration is known as ultrafiltration. Did you know that, on average, the human kidney filters about 1,100 to 1,200 milliliters of blood per minute, which is about 1/5 of the blood pumped out by each ventricle of the heart in a minute? The amount of filtrate formed in the Bowman's capsule of a nephron due to glomerular filtration in the kidneys every minute is called the glomerular filtration rate or GFR, which in a healthy individual is about 125 milliliters per minute or 180 liters per day. The blood pressure in the capillaries acts as a major force in glomerular filtration. Besides, there are certain intrinsic mechanisms in the kidneys that auto-regulate the GFR. The Juxtaglomerular Apparatus or JGA is one such microscopic structure that regulates the GFR. The JGA is formed by cellular modifications between the afferent and to some extent the efferent arteriole and the distal convoluted tubule or DCT of the same nephron at their point of contact. The juxtaglomerular cells release renin whenever there is a drop in the GFR, which stimulates glomerular blood flow and normalizes the GFR. Did you know that of the 180 liters of glomerular filtrate formed per day, the amount of urine released is just 1.5 liters? This is because about 99% of the filtrate is reabsorbed in the renal tubules during the process of reabsorption. Reabsorption is performed by the tubular epithelial cells present in the different parts of the renal tubule through active or passive mechanisms. While substances in the filtrate such as sodium, amino acids and glucose are absorbed by active transport, nitrogenous wastes are absorbed by passive transport. Water is also reabsorbed passively in the initial segments of the nephron. Apart from absorption, the cells in the renal tubule also selectively secrete substances such as potassium and hydrogen ions and ammonia into the filtrate to maintain the pH and ionic balance in body fluids. This process of reabsorption and secretion occurs in different parts of the renal tubule, namely the proximal convoluted tubule or PCT, Henle's loop, and distal convoluted tubule. The collecting duct also takes part in the process. The proximal convoluted tubule is the section of the nephron situated between Bowman's capsule and Henle's loop. The glomerular filtrate from Bowman's capsule enters this tubule, which is lined by cuboidal brush border epithelial cells that help to increase the surface area for reabsorption. The proximal convoluted tubule reabsorbs about 70 to 80% of the electrolytes and water and almost all the essential nutrients and vitamins. It also selectively secretes ammonia, hydrogen ions and potassium ions into the filtrate and absorbs bicarbonate to maintain the pH and ionic balance of body fluids. The proximal convoluted tubule is followed by Henle's loop, where minimal reabsorption takes place. However, the region plays a vital role in maintaining the high osmolarity of medullary interstitial fluid. The descending limb of Henle's loop is permeable to water and almost impermeable to electrolytes, which helps concentrate the filtrate as it moves down. The ascending limb is permeable to electrolytes actively or passively, and is impermeable to water. Therefore, electrolytes pass into the medullary fluid and dilute the concentrated filtrate as it passes upwards. Henle's loop is followed by the distal convoluted tubule, where conditional reabsorption of water and sodium occurs. This region helps maintain the sodium-potassium balance and pH level in the blood by reabsorbing bicarbonate and selectively secreting potassium and hydrogen ions and ammonia. The distal convoluted tubules of several nephrons open into a straight tube called the collecting duct, which extends from the cortex of the kidney to the inner parts of the medulla. The duct helps to reabsorb water, thereby increasing the concentration of urine according to the body's state of hydration. It also maintains osmolarity by enabling small amounts of urea to pass into the medullary interstitium. Further, it selectively secretes hydrogen and potassium ions and maintains the ionic and pH balance of blood. Therefore, the nephron filters blood by reabsorbing substances that are needed and excreting the rest as urine. The urine produced by our body is around 4 times more concentrated than the initial filtrate, which reflects the conservation of water in the nephrons. Henle's loop and the vasa recta are the parts of the kidney that help concentrate the urine. A countercurrent is created in both these parts. This is because the direction of the flow of filtrate in the two limbs of Henle's loop and the flow of blood in the two limbs of the vasa recta are opposite. The countercurrent, along with the proximity of Henle's loop to the vasa recta, maintains an increasing osmolarity in the inner medullary interstitium. The difference in osmolarity between the cortex and the medulla is pronounced, as we can see that the osmolarity in the cortex is 300 milliosmoles per liter, while it is as high as 1200 milliosmoles per liter in the inner medulla. This difference in osmolarity is caused by the difference in the concentration of urea and sodium chloride. The sodium chloride that is transported through the ascending limb of Henle's loop is exchanged with the descending limb of the vasa recta. Then the ascending limb of the vasa recta returns the sodium chloride to the interstitium. In the same way, small amounts of urea entering the thin segment of the ascending limb of Henle's loop are transported back to the interstitium by the collecting tubule. This unique arrangement of the Henle's loop and the vasa recta, together with the countercurrent mechanism, help maintain the concentration gradient in the medulla, which aids the easy passage of water from the collecting duct into the medulla due to osmosis, thus concentrating the urine. Urine thus helps to eliminate the numerous waste compounds generated by our body. Urine formation is vital for human health, and it is difficult to survive without producing and eliminating it.